We're continuing on with topic 3.3, the cell membrane. We're going to get into what is the membrane, how things get in and out, and some of the basics about how cells can kind of control that process. So why do we even care? Right? The membrane is the gateway for anything moving in and out of the cell, and our cells have to be able to control that in order to be able to go through their daily routines. If they had no control, then they would basically be non-living components. So let's dive in where we look at all cells have a plasma membrane. No matter if you are an animal, a plant, bacteria, archaea, protist, doesn't matter, you're going to have a plasma membrane. And the main things the plasma membrane is responsible for is separating the inside and the outside world of the cell, so just creating that barrier, moving food in and waste out, building and exporting molecules, communicating with the environment and other cells, and controlling the movement of molecules in and out, including water. So the plasma membrane has some pretty important jobs, and it goes beyond just serving as a barrier between inside and outside, right? It really is an active component of the cell. So we look at the plasma membrane, right? We usually call it the gatekeeper because it's going to help be that barrier, serve as that control point for the movement of things in and out of the cell. Now, primarily when we look at the plasma membrane, we're made up of a phospholipid bilayer, which we'll talk about in a little bit, as well as these pores, channels, right, gateways that things that can't pass through that bilayer can get through. The main component of the cell membrane is the phospholipid. And the phospholipid is a really cool molecule because it has both a hydrophilic or water-loving phosphate head and two hydrophobic or water-fearing fatty acid tails. And so we have this kind of dichotomy where we have a portion that likes water and a portion that does not like water. So let's look at that. So here's the phosphate head. It likes water. It's a glycerol plus a phosphate. And then the hydrophobic water-fearing or not attracted to water tails, that is mainly a hydrocarbon chain. So it's carbon and hydrogen in a long chain. Now these phospholipids, like we said, they're the primary component. They have this water-loving head, this water-hating tails, and they're arranged in what we call a phospholipid bilayer, where there's two layers with the heads facing on the very, very outside of the cell and the very, very inside, and the tails being sandwiched in the middle. Now, because of this, the membrane is considered semi-permeable. That water-hating middle is going to make it so that not everything can easily pass through. So only certain things are going to be able to pass through the membrane, and so because only some things can get through and other things can't, we call it semi-permeable, right? Partially permeable. It's only permeable to some things, not everything. So let's look at that phospholipid bilayer and what it kind of looks like. So like I said, we have the heads, the water-liking heads, facing the very, very outside and the very, very inside of our cell. And then those tails are sandwiched in between, which makes sense because there's mainly going to be a lot of water outside and inside the cell. And so the heads are going to be attracted to where the water's at. And the tails, since they don't like the water, they're going to try to congregate as far away from the water as possible. Since water's both on the inside and outside, that means they're going to get squished, sandwiched in the middle. The membrane, though, is not this concrete, unmoving, unchanging thing, though. The membrane is what we call a fluid mosaic. We usually call this the fluid mosaic model because these phospholipids and the proteins embedded in with the phospholipids can actually move around, right? And the things that can affect the movement of them could be things like temperature, right? So if there's a really high temperature, things can move around a little more. But if it cools down, the movement kind of slows down with it. But regardless of temperature and whatnot, these phospholipids and the proteins embedded in them are freely able to move around. So if I were to attach maybe a dye or a stain to a certain group of phospholipids and I left that cell alone and I went back and looked at it again, I would find that the dye attached to the phospholipids would be dispersed throughout the entire membrane given enough time. It's not concrete, it's not completely solid to the point where nothing can move, it's not rigid. There's some movement, there's some fluidity to this membrane. Let's look at some of the individuals who hang out in the membrane along with the phospholipids, like receptors. Receptors are able to basically listen or respond to a signal being sent by maybe another cell or from somewhere else in the organism. Recognition proteins basically are they're basically like a name tag. So they serve as a way to identify, hey, what kind of cell are you? Are you from this body or are you from somewhere else? Transport proteins, these guys serve as a pathway for maybe certain molecules to get through either in or out of the cell. Membrane enzymes, these guys are going to help accelerate and speed up chemical reactions both on the inside and outside of the cell. And then up here we have carbohydrate chains. These also are going to serve as a name tag to say, hey, this is who I am, I belong here, or hey, I'm from somewhere else. And cholesterol is going to kind of help provide a little more structure, a little bit of flexibility to the membrane as a whole. So there's a lot of cool components to the plasma membrane. Although the primary component is the phospholipid, right? We see that they make up the most numbers in this picture we see here. So like I mentioned, every cell has an identifying fingerprint. We have to know who is who and whether or not you belong there. It's actually a really important thing, especially in animals, when it comes to your immune system. So every single cell has a different combination of molecules on the surface, including things like glycoproteins, which would be a carbohydrate and protein together, glycolipids, which would be carbs and lipids together, and other recognition proteins. And this fingerprint is unique to the type of cell it is and determines whether or not a cell belongs to the self. So in multicellular organisms like ourselves, where we have lots and lots and lots, hundreds of thousands, millions, billions of cells making up our body, we have to know who is who because not every cell inside of our body is actually from us. We have beneficial bacteria that live with us, maybe things like yeast that live on us and in us. And not all of these are harmful, but we need to be able to tell who's part of the body and who is a guest, so to speak. There's also individuals like certain bacteria that cause disease and illness, and we definitely want to be able to say that they do not belong here. So we have these name tags to be able to say who's part of this body and who is not. So in animals, when a cell recognizes a cell as non-self and not belonging to the body and not basically invited to stay, we get a response from the immune system. The immune system will recognize that cell as a foreigner, as an invader, and it will attack it and destroy it. So this is why we're able to fight off illness, right? You get a cold and your immune system ramps up in response to that cold virus because it recognizes the virus as non-self and not belonging and not allowed to stay in the body.